you can also see how much peptide is there. And based on the concentration of the peptides, you can put the samples into the mass spec, and that you can go for any kind of global proteomics acquisition. So yeah, this is for the insol digestion and the peptide quantification for any kind of
Thank you.
insolation digestion.
Okay, so now I think the samples are ready. So we can try to take the sample and proceed for the mass spec analysis. So TS will now take the peptide and will go shortly to the one of the instrument which we have, which is high throughput proteomics instrument for doing the deep proteome analysis. Most of the time, your goal is first to look at total how many protein can we take from our that sample, right? So in that way, you are now going to do the deep proteome analysis, and there could be different workflow as we talk DDA, DIA. Even within DDA, one could go with the label-based approach or label-free approach. Label-based approach means you have, let's say, multiple conditions to compare, and you can label them with different type of isobaric labels. For example, tandem mass tag or isobaric tagging for relative absolute quantification ITRAC. And you can use these reporter ions and label with those type of set of the multiplex isobaric tag of TMT or ITRAC. And then you can pull them together and you can do the high pH fractionation. And then each of those fractions you can run separately. So that's one way of doing the proteomics, quantitative proteomics. Other was the label-free approach, where you are running every sample separately. And then you are observing their entire chromatogram pattern and hoping that retention time is quite well maintained by spiking in some of the IST peptides, international peptides. And then you are going to compare the data and look at their overall intensity, they're looking at the spectral count, and based on that, you can analyze the data. Or the third approach, as I said, it could be DIE, data independent acquisition, which is the new workflow which is being optimized. So all of this can be utilized. And now TS are going to show the instrument, Orbitrap Fusion, which is one of the tribrid instrument. And we primarily use for a lot of deep proteomic based analysis and quantitative analysis. So Amit and Ankit, please discuss about the Orbitrap Fusion now.
Yes, sir. Thank you, sir. Yeah. So, hello everyone. Good morning, everyone, and welcome to the side session. So, I think Amit has shown, right, like we have taken the samples and have performed the peptide quantification. And now, if we want to do the mass spectrometry analysis, so these are the tubes or vials in which we keep for the mass spectrometry that sir was mentioning about the orbital fusion or the tandem mass spectrometry. But before you know that before going into the mass spectrometry or the MSMS, so it is actually LC MSMS that we generally talk about. So LC here is nano LC or nano liquid chromatography. So you know that what peptides is from the complex samples, like from a tissue, cells, we get it contains a complex mixture of proteins, then which we are digesting it into peptides. Now we want to see that what are the peptides that are present and quantify them, right? So this cannot be achieved if we inject all the peptides at one go, because the detector and the mass spectrometer, it will get oversaturated, and that's why what we need a kind of peptide separation, which we do using reverse phase chromatography. And for this instrument, we use nano LC or nano liquid chromatography. So why it is nano? Because I would like to, so we can focus here. So you can see the flow rate. It is in nanoliter per minute. So where flow rate comes in a nanoliter per minute, so you can understand that it flows in a very small volume quantity over the time, so that the peptides, complex peptide mixtures that we have got, can be easily separated out. Now, this I am to like to show that this contains the two solvent systems using two pumps. Here we make the gradient. So one contains acetonitrile, one contains 0.1% FA water. So with that, the gradient of 40%, 50%, 60%, 80%, all these gradients are made so that the peptides can be eluted as per their hydrophobicity, like in the 2D and the gel molecular weight and pi is used. Here we are taking the another peptide characteristic, that is hydrophobicity, to be used, and these two solvent systems, this chromatography is helping to create the gradient to which it gets eluted out. So these samples that we bring, so what we do is that here is an auto sampler. So just, yeah, so here is the auto sampler where we just put it up, okay, and then we have just eject the tray. So now once we eject it, the tray, so now we can give the command, the instrument to take. Before going into the parameters and those, I'll just like to show like what Sarah has mentioned. So this is the mass spectrometer which is connected to this nano LC. The peptides are getting injected using this column. So this is a pre-column followed by a column. So column is nothing but a pack of C18 material that binds the peptide, and then as per the in the stationary phase, and when the mobile phase solvent comes, so the peptide gets eluted out and gets injected into it. Now, as so you know that now we talk about LC MSMS, so tandem mass spectrometer. So MSMS means there should be two mass specs because we want to get the single sequence or the amino acid that is there. So now this instrument gives, like, there are three mass analyzers present in this instrument. So one is quadrupole, which we need to segregate the first precursor ion, first kind of window where we are filtering it out. Next, it contains two more mass analyzers are there. One is orbit trap and the other is ion trap. So you can either couple quadrupole with ion trap, or you can couple quadrupole with orbit trap. And in between there is a C trap which actually captures the ions and then give then according to the method and the parameter that you give, so it will distribute the ions there. So this gives us an enhanced separation with a superior resolution and a greater select sensitivity. So this instrument gives you a kind of, you can go to a very high 120,000 kind of resolution with a greater speed and accuracy. Yeah. So now I'll just show you something. So yeah, so when you will be doing, so they will be getting this kind of chromatogram. So you can see that if I just try to show you, so it will start from 0 to 120. So 120 means that we are giving a 2-hour long gradient where the peptides are being eluted out. So here the from the we are going from lower hydrophobicity to higher hydrophobicity, and the peptides are eluted out at each phase. So while the peptides are eluted out, it is going to the detector and we are seeing the intensity. So you can see here that the intensity is coming in the range of E9. So the maximum intensity of the precursor that has got is E9. But this is the total ion chromatogram of the MS1. Now, if you want to go and see that a particular, so these are all you are getting are your MS fragments. So for a particular precursor, what are the fragments? So once for any precursor M by Z, that is for a total molecular weight of the peptide followed by the charge and its MS, the fragments. Then the fragments are tried to map with the particular monoisotopic mass of a sequence of a peptide, so that we can get the stretch of the sequence of the complete amino acid which are present in a particular peptide, and then finally bioinformatically peptides are matched with the protein to identify the complete what is there. And these intensity values of the peak are actually led to the quantification and how much proteins are expressed in a particular cell. Yeah, so this is in short about the orbital fusion, like starting from the connected to nano LC. Now we can move on to the next instrument, which is another orbital fusion, but Q exactly. Yeah.
All right, thank you, Ankit. It was a good overview of orbital fusion. So again, mass specs can be utilized for different type of omics-based investigation, not only the proteomics, but also it can be utilized for metabolomics or lipidomics, right? So one can always try to utilize the power of these instruments and use them for multi-omics investigation. And even from the same sample, you can think about whether we can, even try to do this, multiple layers of the sample. One could extract from the protein, one could do the metabolite, one could do lipid, and then try to build the layer-wise information. Of course, people also do genomics on the same sample. So then one could do truly integrated omics, in that way. So while this instrument, what we're going to show you now, which will be another Orbitrap instrument, and the hybrid with the when we can do the things for proteomics as well as for the metabolomics, but we primarily use this instrument along with the UHPLC for doing the metabolomics application. And here we have the quadrupole Orbitrap, that's what is hybrid. unlike the fusion, which is the tribrid instrument, right, which with the ion trap was added over there. Metabolomics workflow is quite straightforward. You can use some organic solvent to extract the layers for doing the metabolite as well as lipids, and followed by acquire the data using this LCM SMS setup, and then do data analysis using different type of databases. So now over to Amit, and he will show you the Q executive Orbitrap mass spec and talk about the metabolomics workflow and data acquisition for that. Amit, you can take over.
So the second aspect that we are going to talk about is the Q exactive, where right now we are doing both untargeted and targeted metabolomics and lipidomics too. So as Sir said, the sample process is quite very straightforward. You will add the chloroform methanol and you will get the layers for both the metabolomics and lipidomics. Now we can see this entire the mass spectrometry and there is the HPLC, the high pressure liquid chromatography is coupled in this instrument. In that system, you see LCMS, here's the HPLC is there, and the model name is Ultimate 3000. So you can see this HPLC quite different from the NanoLC. Okay, so there are 1, 2, 3, 4. 4 different compartments are there. The very first compartment is the column compartment. So here for lipidomics, we use CSH column, and for metabolomics, we use kind of C18 hydroxyl gold bond. Now, one of the very important feature, the technical feature of this column compartment is the thermostatic mechanism. So basically, it provides the passive and active heating of the columns. So for the good coverage and to also to ensure there is no RT sheet of the samples or the metabolites or lipids that you are going to see, you should not compromise with the temperature. So this kind of component will keep the atmospheric ambient temperature for 40 degrees Celsius, and it makes sure that there is no such fluctuation in the ambient temperature. So basically for this kind of columns functioning, the column compartment keep the temperature for 40 degrees Celsius. The second compartment is the auto sampler here. So in auto sampler is essential because of the reproduction automated sample injection in the QE. So you will see there are three different trays are there: the green tray, the red tray, and the blue tray. And these are the different vials where we need to keep the samples. And inside it, you can see the insert here. So basically, in this kind of metabolomics and lipidomics, use this kind of plastic vials. Now, in one tray, you can keep 40 different samples. Okay. And for entire sample queue, there are 3 trays are there. So there is 40 into 3, 120 samples you can keep for the entire sample queue. And one of the most important technical feature is that it has lot of wash pores out there. It will get rid of any kind of the contaminants, and it will also get rid of any kind of carryover if that is coming through the sample when the sample is when the sample acquisition is happening. The third compartment from the bottom is the pump compartment. So the pump compartment, let me open this pump compartment. So it will give us the feature of the pulse-free flow. So it means there will be uninterrupted flow of the solvents, and it will also get rid of any kind of back pressure fluctuation. This is another important thing that you can see. It is a knob. It is basically for the purging system. So basically, whenever you try to give the new solvents for any kind of acquisition, whether it's untargeted or it's targeted, you need to get rid of any kind of bubbles in the solvents. Okay, so basically you can just use these kind of knobs and you can start purging the system. So basically, if they have a purging, you will see all the bubbles are getting out of this particular pipe. Now the final rack is the solvent rack. So in case of lipidomics, you use a quaternary solvent system, and in case of metabolomics, you use any kind of binary solvent system. So in case of lipidomics, you use 4 different solvents out there: the solvent A, B, and C, D. So one of the most important feature of this mass spec is that it can give us the information about the protonated and the deprotonated metabolites or even the lipids. So in case of positive mode aggregation, we need to use a solvent A. That is the composition of the composition of the solvent A is the ACN and the water, and the solvent B, it is the composition of the IPA and the ACN. Similarly, in the case of negative mode, the similar kind of solvent is used, but there is no we do not use any kind of FA, but we add only the FA in case of the positive mode aggregation. Now, Now from the HPLC, we have our one of the most important mass spectrometer here, that is the QE. So as sir said, there are two different mass analyzers. There are two different mass analyzers are there. One is the orbit trap and one is the quadrupole. So the very first thing you need to know that you are in the courses you have already talked about, you have already learned about the what are the hard ionization and soft ionization technique. Within soft ionization, there are techniques like MALDI and ESI. So it uses the API, atmospheric pressure special ionization, which is the ion source, and the mode is the ESI, electrosphere ionization. So in case of MALDI, you will get to know that only single charge ions are generated, but in case of this ESI source, there are multiple charges are getting generated. So with the help with the ESI, there is this entire thing is called Hazy-2 probe. So Hazy-2
Amit, I
probe
think
is
it is going more detail than what time we have. So
okay.
thanks for this demo. I think we should move on to the next concept, which will be the targeted approaches for looking at the proteins and metabolites. So QE, as you have already heard, can do multiple things, and we feel this is one of the very robust instruments for doing multiomics. But of course, once you have identified your good targets, whether it's the peptides, protein coming from the, let's say, Orbitrap technology, or you have identified some metabolites and lipid using the another instrument. So then now you want to validate those, and then you can come to the triple quad based instruments, one possibility, or you can also use Orbitrap instrument for another possibility. There are two different workflow. One is known as the multiple reaction monitoring, which you can use primarily with the triple quadruple instrument. And you can also do the PRM, parallel reaction monitoring, which you can do from the Orbitrap technology. So we are going to now show you how to perform the target analysis. Idea is that you already know your which protein and peptides or the metabolites of your interest. You are only going to provide that list to the software, which now instrument is going to screen. A software Skyline is being primarily utilized for this purpose. And then those selected precursors are being fragmented, and you're going to generate their intensity profile and use that and then try to correlate whether your discovery data and validation data are matching or not. So we are now going to discuss the triple quadrupole based instrument and technology for doing target application. So over to Ankit now.
Yes, sir. Thank you, sir, for the brief overview of the targeted proteomics and the instrument. So just now what I'm going to show you. So I've shown you the future and Amit has shown the QE. So now what we are going to show you, one of the instruments is the triple quadruple base, that is TSP voltage, and it is again coupled with an HPLC. So this is not coupled with nano LC like was the one in the future, but this is what we are. Yeah. So what we are coupled with is like with the HPLC. So why generally what is the main difference between nano LC and HPLC is the flow rate. So HPLC we get the flow rates in ml per minute, whereas I have shown in the the chromatography we need in nanoliter per minute. Since we are here just going with a few target peptides and target metabolites or lipids, whatever be that. So that's why we are using in a high flow rate so that we have a less gradient, we use less solvent, and we can get accurate quantification of those particular peptides. So I'll just show you, this is another model of the HPLC which consists of the Vanquish. So here again you will get the chamber of the auto sampler where you can put up some solvents, and this is the pump system, and this is the column compartment. Now with this column compartment, it is attached the triple quadruple. Now if I going to say what is the major difference between triple quadruple mass spectrometer, so one thing is that, so here we are talking about the different mass analyzers. So we need two mass analyzers. Here just focus on the name, that is triple quadruple. But here there is not that there are three quadruples, so don't get mistaken by the name that it contains three quadruple. It contains two quadruple, and in the middle is one collision cell. So it contains one quadruple. So we have already spoken that the quadruple contains the filtering of the precursor ions. Now there is a collision cell in between, and there is a third quadruple. So how we generally give this kind of instrument? Because this cannot go like you can collect all the whatever is present in your sample in a global way. Rather, we have to give the precursors that will be filtered out in the first quadruple, as well as the product ions after the collision energy collision cell that we are getting to identify in the third quadrupole will also we have to give out. So why this is known as targeted or it is specific and precise? Because we are giving the quadrupole one precursor and its product M by this. So it will capture the exact specific for a peptide and metabolite. So that is the actual, we can say, as a fingerprint for that particular peptide or a molecule and the lipid. So that's why you are very precise. So I'll just show you a quick overview like how it is generally we are done. So this is kind of a, I can show you for a molecule how we measure. So this is done using the Skyline software. So for example, this is one of the compounds. So here you can see that it is the molecular weight, so M by Z of the precursors. Okay, and these are the products. So we had instructed the instrument to capture each of these precursor and its product specific for this particular molecule to identify because it is quite signature to this particular molecule. So now after the data acquisition, you will get this type of graph where you can see this is the detention time. So at 5.5 minutes, again, this is coupled to a C18 column. So where the hydrophobicity is used using the different solvent system to generate, and at 5.5 minutes, you could see that it has, or at a 5 minutes, sorry, yeah, 5.2 minutes, yeah. So it has eluted out and it got the peptide. So in this particular retention time, so you have to see that whether there is a retention time shift or not. So Sam was also mentioning about different kinds of QC checks using different IRT peptides or different kind of standard metabolites. And this is the intensity which is getting around that, what is the peak observed under the area. So since this particular will give you the areas of all these particular transitions, it will give us the quantification that what is the actual intensity. But it is, as I have shown, it is quite specific because we can measure the precursor peptide, precursor product pairs, and that's why it is unique and known as the targeted, and we can exactly precise whatever the list we have identified in global can be validated here. Yeah.
All right.
So,
Thank you, Ankit. So I think, student participants who are available online, uh, what all different type of theory you have studied already. Now you can see their actual practical aspect of it and along with the instrument demonstration. Now what we are going to do, we are going to show you a new technology, which is one of the latest in the field, which is the timsTOF technology, which actually does the 4D proteomics analysis. And fortunately, at our startup in collaboration with Bruker, now we have acquired this technology of timsTOF. And I now request our team at Proteomica to share their video and do a live demo over there to talk about the timsTOF technology, which we think is giving us unprecedented scale of the data. What typically, again, in the proteomics, our goal has been to first get as many protein as possible from a given sample. So using different workflow of DDA to DIA, what we have been trying to achieve was around 5,000 protein from a given typical cell line sample. But now where we have reached with this new technology, almost now 8,000 protein, 8,000 plus proteins very easily, and the group analysis even crossing the 10,000 plus protein. So this is a wonderful, incremental change what we could observe now using the 4D omics technology of the timsTOF. So request Dr. Argya now to show the timsTOF instrument and explain about it.
So welcome everyone. So what you are seeing is the India's first TIMS facility here. So just to give a brief background, you have seen the mass specs which can do a three-dimensional separation right in the form of RT as well as M by Z as well as intensity. This is the first mass spec which is timsTOF Pro, which can do a separation on the fourth dimension. That is based on the cross-sectional area. So let's move on. I will explain the later. So this is what you are finding is the timsTOF Pro instrument coupled with the EvoSep Nano LC. This EvoSep Nano LC is one of the cutting edge revolution in the world of Nano LC. Here you don't have to keep different samples here. You just have to put this kind of tips, which are Evo tips. You directly load the samples and the samples will be directly eluted. Rest all the things are like the general things. You can see the pumps. Through the pumps, once the sample is injected, is brought here. from the sample injection, it goes to the column. Like you can see, this is the column here. So this is a 15 centimeter column which is being attached. This is the column toaster. And now, once the column has been done, it is getting injected into the mass spec. So what happens? You have to think like a ship. Like there is a ship which is sailing. The ship which has longer pals can capture more amount of air and it can move faster. Similarly here, the bigger ions which are there, it will move faster. And similarly, the smaller ions will be left behind. You will have a drag force also. At a certain point of time, it falls down, and then it goes to the timsTOF pro the top detector. So you have to understand there is no exclusion of any ions. All the ions which enters into the timsTOF pro goes into it. That is why its sensitivity and specificity is very high. You can see 800, like you can see like, sorry, you can see like 8,000 proteins with only 200 nanograms of protein. So this is the software, what you can see. This is how the samples are being run, going to the quick elution viewer. So you can find, this is one of the DDA. Let me show you. Okay, so this is one of the plasma serum samples. You can see the chromatograms. This is how the chromatograms look like. And when we have gone to the online protein detection, it can yield, like, just imagine 200 nanograms of serum samples or the plasma samples have the capacity to elute around 1208 proteins, like more than 1000 proteins, which is more than the conventional proteins. Similarly, we can see the HeLa also, like this is the HeLa, the cell lysate pattern. From 300 nanograms of the peptides, we can easily get 7816 proteins, like more than 7,000 proteins. So this kind of cutting edge technology is now revolutioning. The most important thing is like you can store the samples, you can analyze after 6 months. As we have the property of ion mobility and the cross-sectional area, the sample, if it is degrading or something happens, but the areas will be the same. So reproducibility is very high in this kind of cases. Like this is one of the kind of it seems to be.